But I thought it would be worth sort of just highlighting. I'm sure you've all heard this quote by Dobzhansky, and that's who is in the picture there. And we often talk about Darwin, but Dobzhansky sort of articulated this, I think, particularly well for biology. And more recently, there's a husband and wife team, R&P Grant, who played on that same expression, nothing in evolution makes sense except in the light of ecology. And of course, evolution and ecology are intimately entwined. And so when we talk about evolution, we really can't ignore ecology. It's maybe convenient to ignore it, but the fact is that, it's decided to sort of switch itself off. The fact is that one drives the other. So for those of you that don't know IMO, IMO is Integrated Mathematical Oncology, not just in my opinion. And it's a kind of diverse group of scientists. You've met a few of them or heard about some of them. And the whole point of the integration was to put mathematicians inside Cancer Centre and integrate with experimental and clinical colleagues. And one of our sort of main goals is to better understand, predict and treat cancer. And so Joel showed this sort of link that we see mathematical modelling as a great way to sort of integrate the clinic and the experimental aspects of cancer. And up in the 4th floor, which is where I was located, we have Bob Gatenby, who's a total visionary, one of the kind of drivers of evolutionary therapy. We also have had Bob Gillies, who was an experimentalist, an imager, that kind of brought the experimental reality to what we did there up on the 4th floor. And as you know, sadly, he passed away late Tuesday evening, and I thought it was worth just briefly talking about some of the things that he did. I'm not going to spend a lot of time because there's an extensive list, but he published over 400 publications in his sort of 50-year career, and he did some amazing stuff, and he did some of that at Moffitt, but also before. I got involved in looking at cancer metabolism purely because of the work that he did. The idea that tumours are acidic was sort of understood, but he was one of the first to sort of image them, to quantify that, and then to show how it really modulated metastasis, in particular immunotherapy, because the immune system doesn't like acidity. So you can see there's this push and pull that goes back and forth there. And of course, he was the man that defined the field of radiomics and so much more. So he'll be sadly missed. But he had a huge impact on the research that we do there. And Moffitt did a very nice sort of press release where they kind of shared some of this stuff. So it was very nice. Anyway, I wanted to sort of draw your attention to some of the resources that we've been putting together as a community. And so, many of you here are probably a bit more focused on the competition. I don't know why it keeps switching itself off. It's quite odd because I'm not touching it. But there's a kind of bunch of work that's going on in mathematical modelling, in particular applied to different aspects of cancer evolution, but also treatment. This website has a bunch of resources that I ask you to sort of check out, and if you want to hear more, you can talk to Maxie. If you're interested in getting involved and bringing your perspective, we're interested in having you contribute. We actually want volunteers, so please approach us. You've also maybe heard a touch about the IMO workshop a couple of times throughout this meeting. It's an annual meeting that we had for 9 years solid, and then the pandemic hit. And it's not something you can do online. It has to be in person because you're literally brainstorming for a whole week. But it's a lot of fun. It's very intense. It's a competition. Often we have 5 teams of about 10 to 15 players, and each team is led by a mathematician, an experimentalist, and crucially a clinician. And each year we have a different theme for the workshop. And you saw Joel yesterday wearing his t-shirt for the evolutionary therapy one. The theme for this year, we are going to run it this year, come hell or high water, because it's been too many years, and this is going to be a special one because it will be our 10th year. And the theme is going to be cancer communities. So it seems very pertinent to this audience. So think about that. I'll release a kind of application form. We pay for everything. All you have to do is apply and get selected. So you have to sort of justify why you should be picked. We pay your flights, your accommodation, your food, everything, and it's a lot of fun. So keep an eye out for that sometime in August. But let me talk now specifically about the research that we're doing. And I always think it's worth coming back to the reason why we're doing it. This is a patient with metastatic melanoma, and it's a whole body scan, and it's metabolically pseudo-coloured to show where there's a lot of active metabolism. And so you can see that they've got lots all over their lower torso and abdomen. And so if you saw their skin, it's actually black with tumour. It's like a gruesome looking thing. This is definitely a nicer way to look at it. What's kind of very cool is that we developed these drugs, not me personally, but the community, the cancer community, that can target the specific mutations that these tumours have. And within literally, I think it's 4 weeks, 6 weeks maybe, when you look at that person's skin, they look cured. They're actually very healthy looking. They're kind of feeling very good. But as you can see, within just below the lungs there's still some lesions that are in green, and they're not going to respond to that drug. And unfortunately, within 6 months, what happens is that tumour comes back, and it comes back almost in the same locations that it was. And so then that begs the questions, well, did we actually eradicate it, or did we push it into quiescence? Maybe we eradicated some of it, but not all of it. And so our big problem with targeted therapies, amazing though they are, they directly drive resistance. And so one of the things that we've been doing at Moffitt is trying to develop therapies that instead of ignoring the emergence of that resistance, they try and exploit it. So we actually are developing what we call evolutionary therapies, and I'm very fortunate that Moffitt sort of gifted me a centre of excellence, which basically means that we get money to do this research, and particularly we can get philanthropic donations to do this research, and we've been quite lucky on that front. And again, Bob and Damon Reed, who's a pediatric oncologist at Moffitt, are the co-directors of it. And our sort of whole focus there is really to develop and deploy personalised cancer therapy through the integration of predictive mathematical models, patient data, and evolutionary principles. So modelling is actually part of the decision process. That's the whole point of it. You've heard all about adaptive therapy, so I don't really need to give you all the details of this anymore. So I'm basically just going to give you an example of it. The key point here is that one of the main drivers of this is this concept of competitive release. If the tumour is heterogeneous and some of those cells in there are sensitive and some of them are resistant, there's potentially competition between the sensitive and resistant populations. So if you wipe out all of the sensitive, you've released the resistant cells from that competition, and now they grow freely. And so, you know, Bob's kind of genius idea was to sort of play with switching treatment on and off to really enhance that competition. And a few years ago, Jill Gallagher in my lab, and you can go to that GitHub location and play with all the simulations yourself. There's all sorts. Developed this very cool visual, I think, for showing how adaptive therapy works. 2 identical tumours, they're simulations. I'm not going to get into the details of it. You can see it in the paper. But the colour of the cells in these little simulations show how resistant and sensitive they are. So the most sensitive are white, the most resistant are kind of black, dark red. And there's a really hardwired trade-off here. So the most resistant cells divide the slowest, and the most sensitive cells divide the fastest, right? So there's basically, I'm really sort of slanting this towards the sensitive population. And then you can see the distributions of the different populations above. MTD, or continuous therapy, on the right, maximum tolerated dose. Adaptive therapy on the right, sorry, as you face the screen. And what you can see there is that I'm going to switch the treatment on and off with a very simple rule of thumb. That is, when the tumour shrinks 25%, stop the treatment. When it returns to the size it was, start the treatment again. And so when I do this, what you see is very rapidly on the left, we release the competition from the resistant cells, and it grows into this big resistant mass. And on the right, you can see the competition is maintained. We're still trapping those resistant cells. And so this is very much a spatial trapping, right? And of course, I've sort of hardwired it because the sensitive cells will grow back quicker every time I stop the treatment. But you can see there's a kind of yellow clone there that's sort of emerging that's just a little bit more resistant than the rest. And eventually this is going to fail. But I think that's a really great visual for how adaptive therapy works. And of course, one of the things it depends on is that there is a cost of resistance. And so here's some real data from a breast cancer cell line and a couple of melanoma cell lines. The breast cancer cell line there is MCF7, which is a pretty famous one that many people use. And red is the doxorubicin resistant counterpart, and green is its sensitive one. When we co-culture those very rapidly, within about 10 days, the sensitive population, in the absence of drug, of course, outcompetes the resistant. So that shows you quite clearly that the sensitive cells are fitter when there's no drug. Interestingly, if we look at the melanoma cell lines and focus on the WM164 and its resistant counterpart, the blue and the black lines there, that's the targeted drug that they're resistant to, that I just showed you, that sort of cured that patient, or not cured it, transiently cured the patient. And what you can see is that the black is above the blue, so again, the sensitive cells are fitter than the resistant. But if we look at the 1205 one, you can see the sensitive and resistant are actually almost the same. There's no cost of resistance there, right? So what's clear is that sometimes there's a cost of resistance and sometimes there isn't. And a kind of interesting question, which I'm not going to touch on, but we're working on, is how does context modulate the cost of resistance? Okay, this is a kind of more of a maths meeting, so I had to show you a couple of mathematical models, and of course, what better one to start with than the classic L'Octa Volterra. This is some of Max's work that was published, and I believe we have both the lead author, Max's sitting, where are you, Max? Right there. I'm sure you spoke to him. And we also have the reviewer in the audience who sort of admitted that. So that was quite cool. It got accepted. So anyway, this is a very simple competition model, right? You've all heard of it. We've got sensitive cells and we've got resistant cells. We've got kind of a Norton-Simon drug kill. And we have also equal turnover rate, I'll tell you after. And so one of the key things here is that this idea that there's going to be some random death. And that's a question that I think came up earlier in one of the talks, that, you know, how much death is in a tumor as it grows? How much is it actually losing cells purely because of, say, mutation, lack of nutrients, whatever, not because of any drug? How much turnover actually is there? Does anybody have an idea? Guess? Let's say like 10%. Sorry? In terms of birth, yeah. 90? So that's the thing. We actually aren't sure, but some of the estimates were up in the order of 50%. But if you go back to the 60s when people actually measured stuff like this, it could be as much as 100%, meaning literally the tumor's rebirthing itself almost on a weekly basis, which is kind of insane. Yes. So, you know, when you think about that, that could actually have a big impact on how we're thinking about how these tumors are evolving, right, if there's that much death. So here is a very simple comparison with that model where we do continuous therapy and adaptive therapy, and this one here is the 50% rule. And the crucial lines you want to look at is orange is where it fails under sort of maximum tolerated dose or continuous, and blue, the blue dashed line is where it fails under adaptive therapy. And what you can see is there's also lots of other colors here. There's a green line and a red line. Hopefully there's no color blind. Red is the resistant population, green is the sensitive. So you can see you got the classic competitive release there on the left, and then you can see on the right there we have sort of this sort of oscillation on and off, and the shrink and growth of the sensitive and resistant. Eventually it fails too, but you buy a little bit extra time. Now if we do that same adaptive therapy but we increase the amount of turnover that occurs, and here we only go up to 25% turnover, you can see that I'm gaining more and more with adaptive therapy. Okay? But you also see we gain in maximum tolerated dose strategies too, right? So if the tumor's turning over, it's also potentially hurting itself. So all therapies get better. So one of the things that Maxi did was he looked at this then, if we have cost and we have turnover, what happens when you have those two sort of processes occurring at the same time? And cost here, very simply, is impacting your division rate. Okay, so like, if you're paying a higher cost, you're a slower divider when you're resistant. And so what you can see here is, if you look at our sort of 0, 0 versus our sort of, let's say, high cost, high turnover, then adaptive therapy gives you basically indefinite control forever if you're paying costs everywhere, versus with poor control when you've got very low cost, very low turnover. And then we can look at the sort of off diagonal there, and you can see, interestingly, you've got this sort of almost cost turnover trade-off. What, the implication here is you can get kind of similar dynamics with very high turnover but low cost, or very high cost but very low turnover. And so, that's all nice and theoretical, but how does that compare to sort of reality? And so there's this very nice data set which was publicly available for almost like 10 years, and then they took it offline because the web master or whatever thought nobody was interested, which led us for a year to try and pursue getting the data back online, because our journal wouldn't accept the paper if the data wasn't publicly available. It was a complete pain in the arse. Thankfully, we got it back up there and the paper's published and you can go and see it and you can download the data. The thing that's nice about this data is it's not quite an adaptive therapy, but it's definitely some sort of intermittent therapy. And so it's PSA patients, many of them, they're measuring prostate cancer, and it's primary. And you can see the PSA there is the crosses, and then you can see the sort of treatment on and off with the black and white bar. So we took that data and we thought, let's try and calibrate our model to those patients. And then let's look at where they sit in terms of this cost turnover trade-off. Now, obviously, we can't prove that this is what we see, but this is where the patients fell. And what's kind of interesting is that we see this sort of diagonal behavior where a subset of the patients have very high cost but very low turnover, and some have very high turnover but very low cost. And if you look at the sort of lines coming up, and that sort of shows some of the actual patient dynamics for 1, 2, and 3. And if you look very closely there, you can see that in that one column, many cycles are happening for those patients compared to the 3 column, where very few cycles are happening. You basically get one bump, right? So there's quite different dynamics across those patients, and the fit sort of pulled that out. So that was kind of nice. And one of the things that was even nicer was that Maxi used these phase plane analysis for that to get it on the cover of Cancer Research. So that's the first time in the history of cancer research that a mathematical phase plane made it on the cover of Cancer Research. I'm very proud of that. Well done, Maxi. Anyway, so, I think space is a kind of interesting point, and we were looking at a well-mixed system there. So what happens when we introduce space? And so we've done some very simple cellular automata work, and what you can see here is the equivalent comparison looks very similar to the ODE. But when I show you how this CA works, so you've got green cells are sensitive and red cells are resistant, and we're now filling the domain with this, so it's like you're zooming in on part of the tumour. And when the treatment's on, you get the white flashing. Anyway, you can see basically what happens is that eventually in the adaptive therapy, those resistant cells just take over, but you see there's quite a lot of competition in between, right? So it's a very nice way to visualize what I've just been talking about. So we can actually, because now it's spatial and we've got explicit cells, I can quantify in detail the true competition that every one of those cells feels. And so if you look at that graph in the top there, this shows you the number of blocked cell divisions that occurs during the treatments. And if you compare continuous therapy versus the adaptive therapy, what you can see is when the treatment goes off in adaptive therapy, the number of block divisions shoots way up. You see that? And when the treatment comes back on, it goes down again. And when the treatment's off, it shoots back up. So you can see very clearly the competition grows, shrinks as you switch the treatment on and off. Yeah, because it's a stochastic simulation, Maxi run many simulations to generate those. Yeah, so that's the dark lines are the average, and of course, there's going to be variation, right? And that just shows you a couple of examples of that. So what we're able to do now is actually sort of, in a temporal sense, quantify competition throughout the entire lifespan of that simulation. And what was surprising when we first looked at that was there's actually a lot of competition of the resistance cells with themselves, which of course makes sense, right? But it just never made sense. We never thought about it that way till we actually saw it. So you can definitely see with adaptive therapy, the green wobbles a bit and goes up and down when it's on and off. But the resistant cells are actually the bulk of where the competition is. And so that was kind of interesting. Another aspect of the sort of spatial interactions we started to see, and this kind of led to some interesting results, is that depending on how you stochastically see the resistant, let's call them nests or patches, like if they happen to be very close together, or if they're far apart, you're going to get quite different behavior. And you can see here, if we sort of orchestrate that distance, the further apart they are, the time to progression is worse, because you're getting bigger independent colonies growing. There's quite a bit of variation there. And so, you know, one of the things that kind of we realized was that that meant that, actually I'm going back, sorry. I seem to have missed the slide that I needed to go to. Let me just jump to here. Yeah, so adaptive therapy doesn't always win. And what's kind of interesting is that we can see that when there's no turnover, adaptive therapy actually does better generally, if you look at the pie chart. But when there's high turnover, there's actually 15% or so of those maximum tolerated dose therapies do better than adaptive therapy. And so we kind of dug into that, wondering why that was, because we never saw that with the ODE, with the continuum model. You see it only with the stochastic agent-based model. And if we look at the sort of figure up there, one of the reasons for that is what I was saying. Stochastically, you can actually lose one of those nests, one of the resistant nests. And if you compare the continuous therapy to the adaptive, when you've got high turnover, you can see that you've only got one colony that's growing there, whereas in the adaptive therapy we have 3. And that's just by chance, one of those resistant cells died, right? So it's just a sort of artifact of the model, but that could be a reality, right? That could happen. Another thing that was kind of where it seemed to be interesting and fail is that under continuous therapy, you very quickly clear out everything that's sensitive, and you leave behind all the room for those sensitive resistant cells to grow, and they grow like a ball, you know, so you can very nicely get that growth, which normally would be quite slow growth. But what's interesting with the adaptive therapy, you get a much more fragmented sort of resistant population, which when it's competing isn't growing very quickly, but as it grows, the surface area there is much larger for it to grow once the sensitive population wipes out. So you can see the kind of fingering-like behavior that you get in those cells. Let me briefly take you back to here, because of course one of the interesting things is, well, what does that look like in our when we compare to the patient data? And it's a little bit messier, but it's definitely different, and there's definitely different behaviors. So we still see a sort of trade-off there, but of course noise doesn't make it so good. But we started to see some different spatial architecture emerge from these populations. And if you look at the far left there, there's many more sort of resistant cells. And of course, this is through fits, right? So take it with a grain of salt, versus there seems to be much bigger clusters on the right. So, you know, when the cycles are very slow versus very fast, it's more resistant cells versus fewer clumped islands. And we can sort of hypothesize what that looks like. Yeah, you can see the difference in the cycles again, just as we had before. And so we came up with this, what we're calling the carpet patch hypothesis. And it's not very clean, but it's, you know, a kind of interesting idea that if you've got much more diffuse, distinct, resistant populations versus much more clustered or patchy populations for the resistant, you get quite different dynamics. You get sort of rapid cycles when they're more diffuse and intermixed, and you get slower cycles when they're more patchy. And so that kind of leads us to sort of summarize what we think potentially is important for when you're looking at adaptive therapy and why it might be successful or not. So, you know, competition between the cells, so intra-specific competition here, so between the sensitive cells, but also between the sensitive cells competing with the resistant counterparts. So, you know, a very simple cartoon of that reality, but still, I think, a useful one. Okay, so I wanted to speak now in the last part about applying this directly to some animal work that we've done in melanoma. So that WM164 cell line that I showed you right at the beginning, where there definitely appeared to be a cost, what we did was we put this into a xenograft mouse, and that's where you basically inject a lump of tumor cells into the animal, and then you wait and let it grow, and then you start treating it. Each one of these is lines on every one of those graphs is an individual mouse, and you can see that if you have no treatment versus you have continuous treatment, there's actually a lot of diversity between the individual mice. And you know, the funny thing is we're injecting the exact same cell line with supposedly the exact same numbers. You're getting quite a lot of diversity. But the crucial thing here is what you can see in the continuous therapy, within about 10 days, you're seeing the emergence of resistance. So these cells go in sensitive to the drug, but within almost, you know, less than 2 weeks, you've got resistance emerging. So it's very rapid in which they sort of emerge resistance, which makes us think, is it more of a plastic behavior rather than a genetic behavior? One of the other interesting things with these fixed dosing strategies where you switch it on and off, just like 7 days on, 7 days off, the kind of behavior seems to converge a bit between the different animals as you sort of pulse the treatment. And I think part of that is essentially we're telling them when they can grow and when they can't grow, and so it kind of unifies their behavior a little bit. Okay, so we developed a kind of another sort of simple competition model. This, again, play on Locked Volterra in many ways, but there's a crucial assumption in this one, which is when treatment's on, sensitive cells can become resistant, they can switch to that state, and when treatment's off, resistant cells can return to sensitivity. So it's a plastic model. And of course, sensitive cells can also die. And resistant cells generally don't die unless of sort of random death. So very simple sort of competition equations, and then you've got a kind of binary switch in the behavior when the drug is on or off. And so we took that model and we fitted it to all that preliminary data, and generally it does an okay job of matching the dynamics of the continuous treatment and the kind of intermittent. And so with that, we sort of had some of those parameters sort of hammered down in terms of the growth dynamics and so on. And so what we thought was, wouldn't it be cool if we could use this model now to predict and treat a new set of mice? And so that's what we did. And so we took 10 other mice, and I'm showing you here the kind of strategy which we took, and I'm showing you the fits at the very end. And so the strategy is that we basically get, after a sort of initial bolus treatment, which was 2 weeks, we get sort of the growth dynamic or death dynamic on that treatment. And then based on that, we predict forward whether we're going to see a certain growth or whether it's okay to stop the treatment and remain stopped. And then we feed that back to our colleagues doing the experiments. They do that, implement it, then we get the next time point, we repeat the process, right? By the end of it, you've got many points that you're fitting. So as you progress through this, you're getting better and better at predicting the animal because you're getting more and more points on the individual animal. And so that's what we did. And what you can see here is the individual mice and their 2 week bolus up front. Red is when they're on treatment, black is when they're off. And you know, after showing you all that diversity, the kind of treatment strategies look fairly similar, right? There's definitely a kind of 2 weeks on, 2 weeks off type of behavior. But what's interesting is that if you look at the volume, the V on the left-hand side is that's the volume of the tumor at the end of the treatment. For some of them, you know, you're getting literally a 7-fold increase in the tumor size at the end compared to what it started. So even though it's a similar strategy, it didn't respond the same way. But some of them were kind of barely treated. So like mouse 8, for example, only gets one additional little treatment. And of course, ideally, you would want to run this for far longer, but with animals, you're kind of restricted into what you can actually do. The point was that our average behavior was 50% of what the maximum tolerated dose was. And I can sort of show more statistically clearly what happened when we compare with continuous treatment, the kind of fixed 2 weeks on, 2 weeks off, and a kind of model predicted optimal, which was 8 days on, 4 days off, versus our true adaptive model-driven strategy. So you can see it still does the best, but that tumor is growing. It's not going to stop the tumor in the end. It's still failing. It just fails a bit slower. But one of the kind of nice things is that obviously you use less drug and potentially get longer term control. So that experiment was enough for us to sort of drive a clinical trial. And this is an active trial that's accruing. We have 4 patients right now. We're only going to have 10. It's like a pilot study. So there will be no statistical significance from it, but this is to justify going for a much bigger trial. And crucially, this is randomized. And one of the things that's interesting here is that we have 2 drugs, well actually 3 drugs. We have a backbone immunotherapy, and then we have a kind of either continuous or adaptive combination of 2 targeted therapies. And so we're collecting as much data as we can on these patients. We've got ctDNA, we've got LDH, which is a kind of metabolic marker, we've got imaging, and regular sort of serum markers. So it's ongoing. I'll keep you posted. So, somebody mentioned about how many trials is active in sort of adaptive therapy. This is the current list that we have, and you can see that there's 6, but there's actually more sort of almost ready to come out in that pipeline, and some of which you've heard of, some which you haven't. And I've not spoke about it, and neither did Joel, but there's a really cool one that's a sort of extinction strategy, and I'm sure Joel will tell you all about that if you approach him. The first strike, second strike therapies, that's just one of the newest. But I wanted to briefly talk in the last minute, because I know I'm already late, about the very last one there, which is generating novel therapeutic strategies based on an evolutionary tumor board. So the idea that we can sort of bring integrated team into the tumor board. Now, tumor board, normally what the job of the tumor board is to discuss sort of difficult patients focused on a specific disease setting. So there'll be like the lung tumor board, and they'll bring those cases forward and say, like, what do you all think about this patient? How should we treat them? And normally at the table is the pathologist, the radiologist, and so on. But now we also have the mathematician, the ecologist, the evolutionary biologist, and we're all sitting there in the room together. And the idea really is to essentially build a model for that patient's disease, make a prediction about what the best treatment strategy would be, and then give that to our oncologist colleagues to help them make their decision. So we become a seat at the table in that decision-making process, right? And so one of the things that's kind of emerged from this, which was kind of amazing, to be honest, was the idea of articulating, displaying an entire patient trajectory in a single image, which is not something that they normally do, which seems like a crazy idea, right? Of course you would have their whole history as you're trying to make a decision, but they don't. And so this is one of those such graphs, and what you can see in the lower panel is the various treatments the patient's having. And then I'm highlighting only 1 or 2 that are filled in with the line. But each point there is a separate lesion, a metastatic lesion that a patient has. And then you've got the temporal progression of those lesions. Some of them disappear, and that kind of dashed line is below the visible sort of volume that you see on imaging. And so one of the big problems there is, trying to decide what should be included and what shouldn't. But we wanted to at least articulate all the information. You can also see that all of these treatments, there's many drugs that are given together. They're combinations of therapies for short periods of times with breaks. And so do we actually want to model all of those therapies? Like, where's the level of complexity? How can we balance this process? And so one of the things that we decided to do very early on was come up with a very simple generic model to try and capture lesion dynamics. And this is that said model. And crucially, this model is not mechanistic, because of course it sort of buries all the behavior. But essentially we have the tumour and its dynamics, and then we have its kind of effective, let's call it the efficacy, the E. And the efficacy has an ability sort of to go up and down, so it can become more, generally the behaviour is it becomes more resistant over time. But that can also return to sensitivity too. So, you know, a kind of dynamic sensitivity of that population, which effectively just modulates the amount the drug behaves, right? So, you know, all the kind of beauty mechanism that I was talking about and the spatial interactions are out the window, because now we're just trying to make a decision best we can. And so one of the things that we had to do early on was, for every single patient that comes, is try and get any historic data that we can from patients who are treated with those drugs. And there's many, of course, getting the same treatment strategy, but the very next decision is fairly binary for most of those, right? They're not thinking about it from an evolutionary point of view. And so we train our sort of model based on this historic fits, and we try and capture the dynamics that we see, and that gives us essentially a cone, a bounding cone in the patient's behaviour. And so you start getting things like this. So I should say that the kind of solid line there is the current time point we're at, and we're trying to predict the next line, the dashed line, for the treatment decision. And so you can see you've got this sort of cone there of what we think the behaviour is going to do, and then we've got our kind of two drugs potential application that might emerge from that. And I know I'm flying through this, but ultimately the point was that we could show that the cisplatin and the 5-FU was going to be the better option for them to move to next, rather than the other option, or maintaining the current treatment strategy. And so that was really invaluable for the oncologist to just have that, right, based on very sort of crude dynamics and so on. So this is a work that's very, very active and ongoing. It's very exciting, very stressful, because you're making real-time treatment decisions. Fortunately, there's a whole team. Joel's very much part of that, and Mark Robertson-Tessie kind of really leads it, along with our oncologist colleagues. So they're like, everybody's in it together sort of thing, and we've got real clinical buy-in. And so keep an eye out for a preprint on that shortly. It's coming. So let me just conclude by saying, clearly treatment, especially targeted therapies, select for resistant phenotypes. Timing of those treatment strategies matters, and hopefully I've convinced you that space also can be really important. I didn't really go into the multi-drug stuff that we've been looking at, but I think breaking treatments apart potentially and sequencing them, they can also overlap, can be a really smart way to sort of exploit evolution. I didn't talk about evolutionary double binds, but the idea of escaping one drug, making you more sensitive to another, is something that we think a lot about, especially when we're working on the tumour board. Resistant phenotypes may have a cost, but they might not. You can't rely on that, so we have to be very careful there, I think, as we implement adaptive therapies. Our sort of critical piece going forward that we've realised is we need to be able to dynamically and easily measure tumour burden, right? We need a way to quantify what's going on in the patient as frequently as possible. And the reality of patient care in the US is that the insurance companies will not pay for a scan unless it's been 6 months since their last one, right? Now, you can get a blood draw every 2 weeks. So if there's a serum biomarker, that's going to be a pretty amazing way to measure, right? That's why prostate cancer has been very good. But what about breast cancer? What about lung cancer? Ovarian cancer has a pretty nice biomarker too, but not all of those cancers do. So we need to really be thinking about that question. That's something that we are pondering a lot right now. And if there's one thing you take away from this, is that everything that we've seen is that the patient's response should be integrated into their treatment decision. The patient should become their own treatment control. You should look at what they're doing, the dynamics of what they're showing, and respond to it. Don't ignore it. And the problem is with one-size-fits-all sort of therapeutic strategies is that you effectively ignore what the patient's doing just to deliver the same thing over and over again, and it's going to fail. Right? And maybe for the odd patient it's going to succeed. But what's clear to us is that every patient's tumour has its own dynamic, and we need to think about that. Another bonus potentially is that evolutionary therapies can use old drugs that were basically thrown out because they didn't, pass the FDA expected gain in life or whatever. In fact, we could use them in a very smart sequence way to benefit the patient in the long term. And of course, it's a whole team that did this. It wasn't just me. Maxie of Met Unjung did the animal work. Mark Tessie did the ETB stuff. Ina and Kieran did the sort of animal work. Joel and Bob had influenced all this or been involved in it, and I've got a nice collaboration with Philip Maney. And Zenip's the oncologist, brave enough to take on the melanoma work in the clinic. So let me just finish by saying that we have a post-op position available. There's many in the IMO, we're all looking. And if you're not ready now, you might be ready in the future. There's always going to be something there. We'd love to have you. Think about the IMO workshop. And then the final thing is just to say that Darwin did many great things, and he said many great things. And most people think about the survival of the fittest, but I really like this quote from him far better, because I think that's why we're going to succeed with cancer, is because we're collaborating, right? So those who improvise, collaborate and improvise most effectively have prevailed. And I'd like to think that's us. So let's collaborate. Thank you.